Thanks very much for inviting me, and it's my pleasure to talk a little bit about single-cell data and what kind of lessons you can learn about how gene expression is organized in space and in, maybe if I have time, in time. I think this Paris meeting was really great because we got to discuss, and so I think this is now really the first challenge on single-cell data, which I think is a huge computational activity for the next decade to come. So it's nice to be involved in this. One slide of advertisement. We are now moving into this really beautiful new building, which is between wet and dry research. We are 16 PIs right now, but we have funding for more, and it's actually permanent funding. And so here is now the advertisement. So we have open positions for what you call here assistant professors. So I think we endow them better, but okay, and we party also. So, all right, I'll not go into, so my lab produces single-cell sequencing data. We use mainly DropSeq, and we work on making sense of the data, so that's a really exciting time for us. Producing single-cell data by DropSeq, what we are doing is relatively cheap, so the full cost is at about 5 cents per cell. Okay, I'll skip all this and come to the... I want to make one remark, though. Probably everybody knows about, well, we sequence RNA in single cells usually, but there has been a very beautiful paper last year, actually produced here from the New York Genome Center, where they coupled antibodies to oligos, so not fluorophores like in classical FACS, but to oligos. And when you give this to the DropSeq setup, the antibodies bind to the surface of cells. You can put in hundreds of antibodies like this. And then during the procedure, you sequence the oligos which are attached to the antibodies in each cell. So you not only get the RNA expression per single cell, but also the expression of hundreds of antibodies, or how many are bound to the surface specifically. These are our own data, and so it works really beautifully, maybe even better than FACS. And you can do it for hundreds of antibodies in parallel. Okay, talking about gene expression in space. So this is what triggered the challenge. It's a very famous problem in developmental biology model system, the Drosophila embryo. There are thousands of papers published about this. The picture here is at about stage 6, where the cells are cellularized all at the surface of this embryo. So it's a two-dimensional problem. And we know that each cell is unique in the sense of that it gives rise to a different part of the adult animal. So it is actually important to understand the gene expression in each cell. Together with Rob Zinsen, who runs a fly lab, we dissociated this embryo into single cells and sequenced each cell, and had a resolution of about 4,000 genes per cell, which is pretty good because fly has only 12,000 genes, and in the embryo not everything is expressed, so this is pretty good. Now the question, and that is also in essence the dream challenge, is how do you so now you have dissociated cells, you have sequenced them, but of course you want to know where they come from. You want to know this because the position of a cell to a large degree defines its function and actually developmental output. We know that from countless experiments. So you really want to know the position and the relation to other cells in space you have sequenced. So the original idea how to do this was developed by Rahul Satija from the now at the Genome Center, and Arend Marioni. you use in situ. So if you know some messenger RNA imaging in situ data, for example, here for 2 genes, one in red and one in blue, and you now look at your sequence cell and you see that you detect the red gene and the blue gene, then you can infer that the cell must have come from the pink locations, because these are the only locations where both are expressed. So you can see that by using combinations of imaging data, you can actually, for each sequence cell, infer a spatial position of that cell. So this is what we did for the fly case. We came up with our own way of doing that, but this is published. And we were able to create what we call the virtual fly embryo, virtual because it lives on our computer. But you can access this and you can actually do what we call virtual in situ hybridizations. These are now just done on the computer for almost any gene you want. Okay. So we learned some new biology from this. I'm not going to go into this. But in essence, the challenge, and I think also future maybe versions of this, will go into this direction of you have sequence cells, so that's how the human cell atlas mostly operates in many labs, but you want to guess the spatial structures which are actually behind the tissues you're looking at. So I want to talk about what I call a radically different approach to solving this problem. And that is that I think we should be able to reconstruct spatial organization without using any imaging data. So why am I saying this? Okay, so this actually was a major piece of work for me with my primitive programming skills in PowerPoint. It took me about 1 hour to create this object. So what happens if you take your sequence cells and you just place them at random positions on the embryo tissue? You will see something that looks like this when you now ask for expression of individual genes. But we know this is not how a tissue is organized, right? So the genes have some territories on which they live. Space is organized because we know that space is function. So this is wrong, obviously. You don't need to run any algorithm to see that this is a wrong way of putting cells on the embryo. So if you think about it, most ways of putting cells on the embryo, the vast majority will look somehow like this, and so you can exclude a vast number of mappings, and you can ask, actually now the idea coming up, that if you insist that genes are somehow continuously expressed or live on territories, maybe you can actually then organize the mapping of cells to positions respecting this continuity principle, and maybe the solution is actually unique. And so this is work with Nia Friedman, and the key person of this was not only Nikos, who was behind the virtual embryo, but more Nitzan, who realized that this problem, after you map gene expression to a distance space and the embryo tissue to a physical distance space, you can formulate this as a problem of optimal transport theory. And you can actually solve this, she also showed, by linear programming to match this matrix, to align this matrix to this matrix, respecting continuity, or that cells which are close to each other typically are more similar to each other in transcription space than cells which are further apart. I want to say this is a global approach. So you're with this, you're putting cells on the embryo, and you compute in essence a score which is for the entire arrangement of cells on the embryo. So local inhomogeneities are still allowed here. Okay, so stripes, for example, are still allowed by this assumption. And yeah, so that's the assumption. Okay, that's very simple. I'll skip this so you can embed, if you have in situs naturally in the math. It's actually I just want to show you the results. If you take the database, the Berkeley Drosophila database of in situ hybridizations for about 80 transcription factors, and you now dissociate this in silico, so to speak, into single cells, and you run, so you do you do in situ for some genes. Now you run the algorithm on these dissociated cells. You see that you get back the expression actually not so badly. And again, this is using 0 in situs. If you cheat a little bit and you use 2 in situs, it gets better. And when you use 4 in situs, actually it is enough information to reconstruct this perfectly on that level. So these are not the sequencing data, by the way. So again, these are the Berkeley Drosophila database, and in the bioRxiv paper, we show that when you use the sequencing data, you get essentially the main domains out of the data. So there is a lot of spatial information in single-cell gene expression data, and it's a very interesting problem to try to retrieve this information. Okay, we also tested this on other problems like the mouse intestine. I don't have time to really go into this. Also, this is online, but we basically reconstruct this one-dimensional tissue without using any imaging data. Same for the mouse liver. We get very nice results here as well. The algorithm is called NovoSpaRc. Now I just want to remark, do I have 1 minute left or am I out of time? 4 minutes, okay. So I just want to make a conceptual remark here, if I may. So I'll skip all this, although it's very interesting. But maybe I'll tell you one thing. So you might guess that this is a cell cycle because it's round. This is really the cell cycle. So we sequenced here an asynchronous cells from a plate. And essentially by doing a principal component analysis, we see, and it doesn't matter really how many genes you use, you get a circle here. And when you look for markers of the different phases and you mark the genes by this, you see that it's the right progression. So this is really the cell cycle. Now, you see arrows here, and what we used here is what's called velocity of RNA. So this is a Nature paper from, I think, this year from the Karchenko and Linnerson lab. They used the idea that the ratio of spliced to unspliced reads from one gene actually predicts the future expression of that gene if you assume that transcription is a dominant regulator. If you have a very high number of unspliced reads for a gene compared to the number of spliced reads, you expect that these unspliced reads will be processed and will give cause to a rise of the spliced reads. So you would actually infer that messenger RNAs of that gene will go up. You can compute this actually in a Taylor expansion. And you can do this over all genes, so you get then an arrow in gene expression space, which points at the direction in which the cell is marching, so to speak. And if you apply this to the cell cycle, you see, I think, very beautifully that indeed the RNA, the transcription, is driving the cell cycle because the arrows are actually tangential to the circle. It's interesting to philosophize more about this, but I don't have time. I just want to say that in biology, it's always space-time in some sense. So space and time are closely linked. As we know from biology, often the same genes that organize space are also used to organize time hierarchies. And that's famous actually for fly genes. And this has been worked out. But what you can do is you can use our NovoSpaRc approach to say, okay, let's prescribe a donut shape in expression space. So we just say this is our tissue, so to speak. And now we take all the sequence cells and we ask the algorithm to put them in this, with the assumptions I told you, on that space. What comes out is if you take the gene expression, you run NovoSpaRc now, the algorithm I talked about, which reconstructs space. It actually orders the cells, it puts them on this space indeed such that the cell cycle comes out again. Okay? So I think that's very nice. Now, what I'm not gonna show you, but we tried many other shapes, okay, that could in principle reflect the cell cycle or how cells move through cell cycle gene expression space. And in none of them we got the right progression of the cell cycle back. So it seems like that this shape is actually indeed, so there's a totally orthogonal argument to what I showed you before from the data and the RNA velocities, is actually indeed a circle probably living on a maybe a cylinder in space, but the variable part is really this donut-like space. So I think with these challenges to reconstruct in space, you can also think about reconstructing lineages or to understand principles behind lineage decisions. And I think in the next coming years for maybe more challenges, I don't know if there's interest or not, but I think this would be really fun to explore together and to develop the most promising algorithms. So with this, I want to there's not only the Human Cell Atlas. In Europe, we have a consortium that now actually last week got awarded $1 million to build the full proposal, asking the European Commission for 1 billion euros to study single cell science in disease progressions. We are of course friends with the Human Cell Atlas, so this is just also a way to get funding. But to argue that Europe is not always behind. And I think I'll just say that principles are starting to emerge which allow to predict gene expression in space and time. I think that is super interesting, right? So it's not just running some algorithm or, that optimizes something, but we put into the algorithm continuity of gene expression, but you can put in other assumptions and test them, and so you can learn something, understand something. So that's my lab, to which I'm indebted, and some collaborators. Thank you very much for your attention.
Thank you, Nikolaus. Questions?
Thanks. Very interesting talk. So just out of curiosity, the cell cycle example where you use a donut as the shape, have you tried to use a very much elongated donut and whether that worked?
No.
It doesn't work, or you haven't tried?
We haven't tried. But you can, our paper is online, so the algorithm, you can build this algorithm very easily, actually. Well, so it's all public, so you can try out yourself and see. Yeah.
So do you see...
The point is that you could take lineage data and try to actually understand what kind of organization is behind these lineage data. This is what I find so interesting about this kind of analysis.
So do you see a possible application to this computational method to apply it to a piece of human tissue and try to reconstruct where those cells,
yes.
how are they organized?
Yeah, absolutely, yeah.
Very interesting.
Yeah. That's the aim, right? So with the embryo, we can do the zebrafish embryo, liver, intestine, so we are trying to move into 3D, but 2D starts to work.
So I have a algorithmic question and biological questions. You find a single solution. How hard did you have to work to really converge, and does it mean something in terms of development, in terms of stability of these solutions?
Yeah, so in principle, the optimal transport theory mapping, then you use a gradient descent method, and had long discussions with Moore about this. And, I'm not a computer scientist, but what she tells me that with this mapping, it should converge. Okay, so this is, if I understand her correctly. For the fly embryo, actually, you can choose, so with 4 images, and pretty much regardless of which 4 images you're using, actually, you always arrive at the same solution. This I can tell you. So, and that's already, 6,000 cells, positions is an enormous combinatorial problem to solve. So I was totally floored to see that actually these kinds of problems can at all be solved. And it was new to me. Well, no, we haven't done this, but this actually can be done because there are mutants available, and so you can. There are also data available, so one can explore this. We just did not have opportunity to do so.
Nikolaus, one question. There is some technology that is emerging. I think that there was at least one paper in Science a couple of years ago in which they are able to do a micro dissection of each cell, somehow keep the information of the position and sequence that. So in a way, you know the single cell and you know the position. It will be interesting to see whether your algorithms recapitulate the original tissue there.
Yes, so I think this will be, you're absolutely right. And actually, my lab, we had a poster recently. We have our own way of doing this by using a grid that we put physically on tissue and then label the RNA and so on. So these experimental methods are currently really far from perfection. So you get usually shallow data. Oftentimes you also have to target specific genes, but it's of course improving. There's a lot of investment in this, and yes, so these data will help to see how much you understand really about the single cell data. I think for quite some time, the dissociation will still produce the far deeper data which you also need. But yes, so I think one should use these data and combine. But my real interest in all this, I have to say, is not in how do you best compute this or which method does it best. What I think what is emerging is, in some sense we knew this, but that there are genes which are important for organizing space, right? Which genes are these? What are the principles maybe behind how they actually do the job of regulating spatial organization? And I think these questions are related to also time development. And so I think we are progressing towards a scientific maybe understanding of making sense of these principles. Maybe.
Thank you very much.